The nitrate test, testing for nitrate reduction. The nitrate test is a simple broth inoculation. For this experiment, we will be inoculating 4 different broths. Your instructor may also have you inoculate 1 nitrate broth with some soil. Many soil organisms are an important part of the nitrogen cycle, and the enzymes nitrate reductase and nitrite reductase are used by many organisms in the conversion of nitrogen into various forms for use by plants or other organisms. Shown here is the simple broth inoculation used for the nitrate test. Make sure to flame your loop in between each bacteria. Once the broths have been inoculated and incubated, you will go through 2 different steps to interpret your results. In step 1, you will add 2 reagents, nitrate A and nitrate B. After the addition of nitrate reagents A and B, if the broth changes to the color red, it is considered positive for nitrate reductase. If there is no color change in the broth, the test is considered inconclusive, and you must move on to step 2. In step 2, you will add zinc powder and hydrochloric acid. At this point, if the tube turns red, the test is considered negative. If there is no color change, the test is considered positive. Nitrates in the media are broken down by nitrate reductase into nitrite. If nitrite is present in the tube after incubation, the reagents A and B will detect it. The presence of nitrate reductase only will be detected by the reagents causing the media to turn a bright red. Here you can see the media turning red after the addition of both reagents. For the next tube, reagents A and B will be added as well. If the bacteria produced nitrate reductase, the nitrate in the media will be converted again into nitrites. However, this tube does not change to a bright red after the addition of the reagents. This is due to one of 2 possible reactions. The first reaction would be no production of nitrate reductase or nitrite reductase, a second enzyme produced by some bacteria. If no enzymes were produced, this is a non-nitrate reducing bacteria. In this case, the entire test result would be negative. However, some organisms are capable of producing a second enzyme known as nitrite reductase. Nitrite reductase will reduce nitrites into nitrogen gas and nitrous oxides. If nitrogen gas and nitrous oxides are present in the media, there will be no nitrites for the reagents to detect. Here, zinc powder and hydrochloric acid are being added to the tube after the addition of nitrate reagents A and B. Carefully swirl the tube to mix the contents. Allow them to sit for up to 2 minutes to see if a color change occurs. If the reagents turn red, this means that hydrochloric acid and zinc catalyzed the breakdown of nitrates present in the media. This means that no enzymes were produced by the bacteria, so this test would then be negative. So red, with the addition of zinc powder and hydrochloric acid, is negative for nitrate reduction. If there is no color change and the media remains a clearish brown, this means that both nitrate and nitrite reductase were produced, and the final products are nitrogen gas and nitrous oxide. In this case, the organism produced both enzymes. The tubes shown here have both had reagents A and B added, as well as zinc powder and hydrochloric acid. The tube on the left is negative, and the tube on the right is positive. The tube on the left has no enzyme production. The tube on the right has both enzymes being produced. A key way to tell the difference between a red positive and a red negative is by the presence of the zinc powder in the bottom of the tube. Now shown are the 2 possible positive reactions. The tube on the left is positive. It turned red after the addition of reagents A and B. If the tube turns red after the addition of reagents A and B, no second step is necessary. The tube on the right did not change color after the reagents A and B, so zinc powder and hydrochloric acid were added. The tube still did not change color, indicating that no nitrates or nitrites are present in the tube, so the organism grown here produced both nitrate and nitrite reductase. This flow chart shows the different possible reactions depending on which or if both enzymes have been produced. So remember, some organisms may produce nitrate reductase only, others may produce nitrate and nitrite reductase, and yet other organisms will not produce either. And using the nitrate test, you can tell which enzyme is being produced. Nitrogen is an important element for life, and many bacteria play key roles in the conversion and fixation of nitrogen into different forms for use by plants and other organisms. In addition to agricultural applications, nitrogen fixation or nitrate reduction can also take place in clinical applications, and the nitrate test is important in the classification and identification of many gram-negative and gram-positive organisms.